Our next speaker is Dr. Amy Rosenberg of CDER, FDA in Bethesda, Maryland. Amy, you may go ahead and start sharing your slides. Dr. Rosenberg received her MD from Albert Einstein College of Medicine and is board certified in internal medicine. She was a postdoctoral fellow in Al Singer's laboratory in the NCI before coming to CDER FDA. She became director of the Division of Therapeutic Proteins, now DBRR3 in the Office of Biotechnology Products, CDER, which regulates diverse protein therapeutics, including monoclonal antibodies and fusion proteins, enzyme replacement therapies, hematologic and somatic cell growth factors, and immunomodulatory agents. She has been a driving force in risk evaluation and mitigation pertaining to the immunogenicity of therapeutic proteins, guidance for the mitigation pertaining to the guidance for industry, immunogenicity assessment for therapeutic protein products in 2014, and in the elucidation and implementation of immune tolerance induction protocols in clinical settings. She's co-edited the AAPS Springer book, BioBetters, Protein Engineering to Approach the Curative. And she'll be discussing immunogenicity in the time of COVID-19, clinical context for reappraisal of immunogenicity of therapeutic proteins. Amy?
Great. Okay, so this is a bit of a switch from the talks we've already heard, and this is concerns actually the protein therapeutics that are being used to treat COVID-19 and what might be their fate in the context of COVID-19. Here's my the typical disclaimer. Okay, so I think everybody's well familiar with the portrait of the immune response and in the hyperinflammatory response in COVID-19. I think, you know, if you look at the clinical symptoms and signs in the early stage, there is a lack of a type 1 interferon response, which is thought to be critical in allowing the virus to spread. And although there are mild constitutional symptoms, nonetheless, lymphopenia is already an issue in the early phase. And of course, this, in the absence of being able to contain the virus by interferon responses, one gets spread to the pulmonary tissues, and then the inflammatory response ensues, which is severe, with elevated cytokines, inflammatory cytokines, and inflammatory markers. And so the real question to be asked here is, does the hyperinflammatory state associated with COVID-19, as well as other entities that it actually mimics quite well, increase the risk of immunogenicity of a therapeutic protein product? And so what you can see here is the inflammatory cytokines and chemokines and markers of tissue and cell damage in SARS-CoV-2, and it strongly resembles that which we see in HLH and in macrophage activation syndromes. Of course, IL-1 beta, IL-6, IL-8, 9, 10, interferon gamma, TNF, and as well as a host of chemokines and these well-known markers. And of course, the immunomodulatory approaches to the widespread damage that's induced in the context of this hyperinflammatory response have to do with treatment with protein therapeutics, with type 1 interferons, with antibodies, monoclonal antibodies to IL-1, IL-1 receptor antagonist, antibodies to monoclonals to IL-6, to interferon gamma. So I think it's well understood that, you know, we do a great job with keeping therapeutic proteins well preserved and stable ex vivo, but we really have very little knowledge of what happens to them in vivo. And it may have actually a substantial impact on immunogenicity when you go from, very low freezing temperatures to 37 degrees and a pH of 7.4. One might expect to see enhanced product aggregation, altered antigen and presentation, alteration of sites and extent of protease activities, generation of novel epitopes, and enhanced HLA binding of product-related peptides particularly from deamidated and citrullonated proteins. And it is actually quite well known that these sorts of changes can occur in vivo. So here's an example of extensive and rapid deamidation of a monoclonal antibody in vivo. And the effects on activity and immunogenicity here are unknown. Probably would not make it into the clinic given this profile. So moreover, we know neutrophil defenses, and there's a very prominent neutrophilic response in SARS-CoV-2, but it's also clear from previous work that neutrophil defenses, as well as diabetes, also a prominent risk factor for SARS-CoV-2, aldehyde tag proteins. So reducing sugars and lipid peroxidation in activated neutrophils release aldehydic products that tag lysine residues of target proteins, which impair protein function, cross-link proteins to generate aggregates, and enhance uptake of these proteins by scavenger receptors into macrophages, and enhance immunogenicity. So we also know that self-tolerance is broken by aldehyde modifications of a self-protein. So here's an example of a glycoaldehydic tagged cytochrome C, which is a fairly well-tolerated self-protein. And you can see that the glycoaldehyde targeted cytochrome C induces a robust immune response. So the question is does the hyperinflammatory state associated with COVID-19, as well as other entities that I had mentioned, increase the risk of immunogenicity of a therapeutic protein product? And there is one caveat, even though you know, this concern about the hyperinflammatory situation and what it might do to immunogenicity, given the profound lymphopenia and immune dysregulation associated with COVID-19, it is also possible that the incidence and or robustness of anti-drug antibodies will not differ substantially, or perhaps even will be decreased from that in other patient populations. So in seeking some clues here, I thought it would be reasonable to look at immune responses to self-proteins in the context of COVID-19, because that might tell us something about administered drugs. So, is self-tolerance broken by the hyperinflammatory response in COVID-19? And it is well apparent that SARS-CoV-2 infection precedes the appearance of various autoimmune and autoinflammatory diseases. And I think we all know very prominently the pediatric inflammatory systemic syndrome, or MISC. And so it has become a considerable concern. And what's the nature of its link with autoimmune as well as autoinflammatory sequelae? And I think it's important to distinguish autoimmune from autoinflammatory, autoimmune being a highly specific response to key antigens, whereas autoinflammatory doesn't necessarily imply such autoimmunity. So what are the molecular mechanisms that trigger autoimmune and or autoinflammatory responses? What are the genetic predisposition factors implicated in the pediatric inflammatory multisystem disease? And a reasonable question is, are autoimmune or autoinflammatory responses associated with any particular variant forms of SARS-CoV-2? What is the nature of the SARS-CoV-2 pathogen-associated molecular patterns that might precede triggering of these responses? And are the mechanisms implicated in the induction of high concentration of IL-6 in kids and adults similar or distinct? So SARS-CoV-2 certainly appears to increase immunogenicity of some self-proteins. And there is a noted increase in the incidence of autoimmune diseases. I want to point out, this isn't necessarily that different from what happens following severe other viral infections, not surprisingly. So autoimmune disorders have been known to increase following SARS-CoV-2 infection, autoimmune thrombocytopenia, the Guillain-Barré syndrome, the antiphospholipid syndrome, as well as autoimmune hemolytic anemia. And the mechanism isn't exactly clear. It could act as a direct trigger of autoimmune and autoinflammatory conditions, either by molecular mimicry, which is doubtful. But the second possibility is that the inflammation and dysregulated immune responses following this infection might prompt other environmental insults to cause the observed pathologies in predisposed individuals. So here's an example of the induction of the antiphospholipid syndrome. These are 3 patients who developed this, and one can see very specific antiphospholipid antibodies, including the anti-cardiolipin IgA and anti-beta-2 glycoprotein 1 IgA and IgG. I think it's interesting that the IgAs are prominent in this, but nonetheless, these patients had multiple cerebral infarctions associated with this, this disease. And additional autoinflammatory disorders and autoimmune disorders are associated, such as the Kawasaki-like disease, Kawasaki disease shock syndrome. Myocarditis has been observed in this, as well as in the macrophage activation syndrome. Compared with classical Kawasaki disease, patients with COVID-associated Kawasaki disease were older, had gastrointestinal and meningeal signs, and they also had leukopenia with marked lymphopenia, thrombocytopenia, increased ferritin, and markers of myocarditis. So quite distinct, and I think really pulling in this idea that there are specific autoimmune targets. And there is a noted high incidence of myocarditis in all patient populations and cardiac involvement. What is behind that? I don't think, to my knowledge, nobody has identified specific target antigens in the heart, but I think those should be strongly looked for as you know, as a matter of routine interest, because it's going to be key to trying to address therapeutically the incidence of this myocarditis. Okay, so I think the answer to our question, based on immune responses to self-proteins, is that the hyperinflammatory state is significantly likely to increase the risk of immunogenicity of therapeutic protein products. So to understand what is of primary concern, I think we go back to the guidance for industry that came out in 2014. And that guidance focused really on the consequences of immune responses to therapeutic proteins as they impact the safety and efficacy of high-risk therapeutics in the context of COVID-19. So we're most worried, of course, about life-saving and potentially life-saving agents and therapeutic counterparts of non-redundant endogenous proteins, as well as immune responses to highly effective therapeutics, such as we're hoping to encounter further in the context of treatment of COVID-19. Just getting to these areas of concern, first of all, in terms of anaphylaxis, I have not seen evidence of increased incidence in COVID-19. I think that's something really to watch out for, but I have not seen reports on that. So cross-reactive antibody-mediated neutralization of endogenous proteins, this really concerns endogenous factors with non-redundant functions such as EPO and thrombopoietin. Neutralizing antibodies to life-saving therapeutics are the other prominent worry. And again, there's the potential for monoclonal antibodies to SARS-CoV-2 spike, RBD, to be a source of anti-drug antibodies. For those who don't remember the 90s, just to point out that thrombopoietin was introduced with the idea that following chemotherapy, it would rescue platelet production. Thrombopoietin is a endogenous factor that is the sole factor mediating platelet production. And so the antibodies, so when this was first tested, the antibody responses to the thrombopoietin and to the next generation MGDF caused severe thrombocytopenia in patients, and it lasted for over a year. The second example was pure red cell aplasia that was induced by antibody responses to recombinant EPO. Now, EPO, erythropoietin, is the sole factor mediating red blood cell production. And so following multiple changes to a recombinant human EPO product in which aggregates became prominent, the incidence of pure red cell aplasia really increased dramatically. We can't forget that this has significant potential for other proteins. So for instance, GM-CSF is being considered for use in vaccines as an example, and antibody responses to GM-CSF are a prominent feature of pulmonary alveolar proteinosis, which is an autoimmune disease in which antibodies to GM-CSF are prominent. Moreover, the potential, there's an additional potential for an immune suppressive product, a CTLA-4 Ig, if antibody response were to develop to that product, and specifically to the CTLA-4 moiety, that response would potentially act as a checkpoint inhibitor similar to ipilimumab, an anti-CTLA-4 monoclonal. Okay, so this is what we are concerned about. And here, for those who don't remember, that for some enzymatic therapeutics, they're life-saving. And this is the example of Pompe disease, in which the enzyme replacement therapy, recombinant human alpha glucosidase, is key in mediating survival, and antibody responses, as you can see here, actually cause patient death. And in fact, this is further proven using Koch's postulates by prophylactic immune tolerance induction protocol for high-risk patients who were given the enzyme replacement therapy in the context of rituximab and methotrexate. And these patients have suppressed immune responses. They're actually immune tolerant, and they have a much better profile of survival than do patients who are not tolerized and who develop antibodies. So when one goes through immunogenicity risk assessments, there are biologic and disease factors, which are listed here. Is there an endogenous homologue to your therapeutic? Does it have a redundant or unique biology? If the endogenous protein level is high, it's much less likely to induce immune responses. Insight from the autoimmune profile, as well as knockout mice. The intended disease IND, is it not life-threatening or is it life-threatening? Here we have SARS-CoV-2, which is clearly a life-threatening disease. And whether there are really other treatment options. Moreover, the patient, product, and protocol factors are important in thinking about immunogenicity testing for products in the context of COVID-19. What is the dosing frequency? Is a single dose is much less likely to induce an immune response than chronic or intermittent dosing. Very high doses are less likely to induce immune responses than low or average doses. Route of administration may or may not be important. Patient immune status, these patients in SARS-CoV-2, they have an activated state, if not a hyperinflammatory state. And so the bottom line is one might expect some, they're more likely to generate immune responses, as we have mentioned, Moreover, the immunomodulatory action of the therapeutic you intend to use. Is it immunostimulatory versus immune suppressant? And certainly for the hyperinflammatory response, I think we're looking at mostly immune suppressant with a lower likelihood of inducing anti-drug antibodies. Okay, so we're going to finish up again, reminding us, you know, in considering testing products that either have been on the market or new products, what are the consequences of neutralizing the product? Again, the safety issues I have mentioned before, the homology, degree of cross-reactivity to human sequences, again, the therapeutic counterparts of endogenous proteins, neutralization of life-saving therapeutics, and non-human proteins, which already are going to be immunogenic, maybe more so in COVID-19. Whether these, your therapeutic is a first-in-class. Now, first-in-class, you definitely want to have a very robust, I believe in, you know, testing for immunogenicity program versus a Me Too product in which you might have substantial knowledge already. You want to be sure your quality control strategy and stability profile is in reasonable line, considering you're going to be putting these products into a hyperinflammatory milieu. Is it an approved drug, or is it a new delivery route? And there may be differences in immunogenicity profiles based on the delivery route and the impact on product quality profile of that new delivery route. Okay, so finally, considerations for immunogenicity testing. Please consider basic risk assessment paradigms. The consequences are primary. Protocol issues are key. A single administration versus recurrent dosing and dose escalation, and consider whether active testing is really required and necessary versus collection of samples to test following the conclusion of the study. Again, risk dependent. Is this a novel agent with no previous immunogenicity data that you're putting into a hyperinflammatory context? I think that's very important in real-time immunogenicity data. Is this a repurposed agent? Is this informative for the repurposing and the original context? Is it informative for immunogenicity testing in this new setting? And this will depend in part on the patient population previously treated. Were they immune suppressed versus immune competent versus autoimmune, as well as the dosing schedule. And I'm going to conclude with that and thank my colleagues at FDA for helping me with this talk, and I'd be happy to take questions.
Thank you very much indeed, Amy. Thank you for your talk. It's great, really interesting. We do have a question and time for one. So Tim Hickling, now from Roche. Tim, would you like to ask a question verbally? I will unmute you so you can speak.
Okay, yeah, thanks, Jeremy, and thanks, Amy. Just was wondering, do you see that with anti-SARS-CoV-2 MABs, where they're being developed, are they taking into account suitable immunogenicity mitigation strategies given this sort of higher risk of inflammation that you're talking about?
I think it's certainly worrisome. I think it's worth real time or you know, very rapid testing, especially if they prove to be highly effective. And as well, you know, if you have the worry of antibody-dependent enhancement, and whether that is likely also to boost the immune response to the protein therapeutic. So I think you have a number of worries with monoclonals. You certainly would not want to see neutralizing responses to a very highly effective therapy very rapidly.
Thank you, Amy. That's great. We do have one more question from Hailim Cho. Hailim, can I ask you to ask your question verbally as well? I'll just unmute you.
Oh, hi. Yeah, thank you. Yeah, thanks for the talk. So my question is, would you recommend conducting the cellular immunogenicity study at the preclinical space using the human PBMC? And if so, in the case of the Pompe disease that you mentioned, especially those patients, like for example, like gram-negative status, would you then recommend using the patient PBMC instead of the healthy donor PBMC?
Yeah, I don't know that PBMCs, outside of the context of the a real hyperinflammatory response are going to be particularly helpful. I guess if you took the PBMC from patients with the hyperinflammatory response, might you gain some knowledge there? Perhaps. I really don't know the answer to that. I'd like to see some studies.
Thank you, Amy. That's great.